How do phospholipids form into bilayers to create cell membranes? So let's take a look at the phospholipids making a cell membrane. We know phospholipids are amphipathic, right? This is a word that means they have both hydrophobic and hydrophilic regions. And so a phospholipid, usually drawn like this, has a polar head, and it has non-polar tails. The non-polar tails cannot interact with water. They just can't. It's a physical impossibility. And so that means they have to hide. So in the presence of water, the phospholipid tails all hide together, and the polar heads, which can interact with water, form on the outside. So you get this bilayer. It's because of its amphipathicity, if I may use that word. Okay, what does the fluid mosaic model of a membrane mean? Don't overthink this one, okay? The words say it all. A mosaic is a mixture, right? The fluid mosaic is a fluid mixture. And so it's really just referring to this diagram here. In fact, I don't know figure 5, well, that might be an old edition. I'm hesitant to put that number there, but anyway, this is the fluid mosaic, which means that these proteins which are embedded in the membrane and these carbohydrates which are attached to lipids and the lipids themselves, these things can move around, right? This is not a rigid structure like a skeletal structure. This is fluid. And so the fluid mosaic is a fluid mixture of components in the membrane. Nothing more to it. Okay, be familiar with osmosis and diffusion concepts. So let's take a look at some of those. Let me read the next couple of things here because they're related. Determine which cells will shrink and which will swell under different osmotic conditions. So let's look at osmosis first, then we'll come back to diffusion. I don't know if we really have too many slides about osmosis. I guess we do. So osmosis, remember, is the movement of water in relationship to the amount of solutes on either side of a membrane. So water, so in terms of osmosis, water moves into the compartment with the highest solute concentration. Okay, this is the first and last thing you need to know about osmosis. It's going to go, the water is going to go where the solute is highest. So, for example, if that drawing's not great. I like my little beaker drawing better. If we have 3 different beakers, and these 3 different beakers are home to our gigantic pet cell, depending on the solute concentration inside and outside of the cell, we can predict where water will move. So, the first one, let's just start easy. If the solute concentration is equal inside and outside of the cell, what do we call this concentrate or this solution? Isotonic. Okay, so in isotonic solutions, water still crosses membranes, but in the end, you break even, meaning that water can go in and water may flow out, but there's no net movement. So when it's all said and done, however much water goes out has also come in. All right, you end up right where you started. Now, what if we have a solution like this? The solute concentration, whatever solute that may be, bless you, is much higher on the outside. What kind of tonic is that? Hyper, right? Hyper meaning too much or more. And so in this case, and this is a reference, remember, to the solution, not the cell. It's a hypertonic solution. So in this case, our rule, water moves into the compartment with the highest solute concentration. So is it inside or outside of the cell where solute is highest? Outside, so that's where the water moves. And so maybe the easiest way to think of it, if the water is moving out, the cell is going to shrink. Okay? And then our only other possibility is that the solute concentration is much higher inside the cell. And so in this case, the solution has less solute, so what kind of solution is that? Hypotonic. Okay? And so in this case, water is going to move into the compartment with the highest solute concentration. So water will move into the cell, so the cell will swell. Cell swells. Okay, so that is a good osmosis concept to be familiar with. Determine which cells will shrink and which will swell under different conditions. See board. All right, now let's go back and look at diffusion. Actually, no, sorry, this is not in a great order. Let's look at the transmembrane proteins, then we'll bring up diffusion and transport after that. So transmembrane, peripheral, and there was another one. There should be. Where's my clicker? There should be a lipid anchored protein as well. So our three main types of proteins that we may find associated with the membrane, we're going to see these for the rest of the semester, and anytime you're studying biology, we're going to be thinking about proteins embedded in membranes. So make sure that this simple three-part concept is in your wheelhouse, as they say, a term I've never understood. What is a wheelhouse? What does that mean? Is it where you make wheels? I don't know. I don't understand it. So there are three proteins, two on this review, but there's a third we should include since it should belong there. But a transmembrane protein crosses the membrane. Usually transmembrane proteins are made of multiple individual, what's the right word? Well, they're called alpha helices. I was trying to think of a simpler word for it, but they're just alpha helices. So these cylindrical structures go back and forth across the membrane. Remember, a protein, when it's made, or if we took this protein and pulled on the ends, we could straighten it out, right? It's just a polymer of amino acids. But when it's in a membrane, it crisscrosses back and forth across the membrane. This is a very, very common protein that we encounter in our cells. Okay, so many different proteins that affect your biology are like this. All right, and so these proteins pass back and forth across the cell. Other proteins, so this is a transmembrane protein. Other proteins that are affiliated with the membrane but don't cross the membrane include the lipid anchored protein. So over here we've got a protein which does not cross the membrane, but it's covalently linked to a lipid, and a lipid is hydrophobic, and hydrophobic structures can't be exposed to water. So that means it's going to be hiding among its phospholipid friends with its fatty acid tail embedded in the membrane. As such, it is a lipid anchored protein, not crossing the membrane, but still associated with it. And then the third one is affiliated with a transmembrane protein, but it's not actually connected to the membrane, right? And if you think about the word peripheral protein, you'll see within that the word periphery, which means around or the outside of. And so the peripheral protein is around the membrane, but it's outside of the membrane. And so this guy is the peripheral membrane protein. Another place you probably use the word peripheral is to think about computer peripherals, things like the mouse and the keyboard. They're not actually the central processing unit, but they are peripheral to it. Okay, so that's important. What are the hydrophobic and hydrophilic sections of the protein? So again, to interact with the membrane has water on the outside and water on the inside of the cell. This was our cell. But the membrane has hydrophobic and hydrophilic regions. The hydrophilic are the polar heads on the outside, and the hydrophobic are the fatty acid tails on the inside. So if you're a protein and you are in both worlds, okay, you have regions that are, by definition, if they're interacting with water, they are hydrophilic. Right? But you have regions that are also crisscrossing the fatty acid tails in the amphipathic phospholipids. And so the regions that cross through the membrane part of the same protein, right? These regions are hydrophobic. So a transmembrane protein, just like a phospholipid, is amphipathic. It has regions that are hydrophobic and it has regions that are hydrophilic. You will want to know that. Okay, so we've got those guys, and then taking up solids or particles, and then diffusion concepts. Let's hit those two things. So in terms of transport, we have transport. Actually, I think we've got this all on one slide. Here we go. We have transport that requires energy and transport that doesn't. If transport requires energy, that means you're doing something that is not natural for those solids, meaning you're going against the gradient, right? You're going uphill, you're doing work. And so active transport that requires energy is taking solids from a low concentration place and moving them into a high concentration place. This is against the natural order of things, so you must use energy to force this process. And in addition to energy, you're going to need some type of transport protein. Okay, they're not just going to move across the membrane to a more concentrated state on their own. You force the issue using energy and a protein transporter. Now, on the other hand, the more natural movement of solids would be to go from high to low concentrations. This is also going from high to low concentrations. The difference being between diffusion and facilitated diffusion, which are both passive, right? They're passive because no energy is involved. The difference is diffusion in general means that, or without the word facilitated, means that these molecules are able to cross the membrane on their own. So these could be small molecules like oxygen gas or carbon dioxide. These guys can move back and forth across the membrane, so their movement is governed by the concentration of that solid. So they go from high to low. In the case of facilitated diffusion, same thing is happening, only these molecules can't cross the membrane on their own. So this might be like a charged ion. It's going from high to low concentration, but to cross the membrane, it needs help. Okay, so it's facilitated, but it's not using energy. Now, in the case of active transport, we do have more than one kind of active transport, so let's look at those guys. Here we go. We have active transport that we would call primary and active transport that we would call secondary. The difference between these two, illustrated in this diagram, is that primary active transport is using energy in a transporter protein to create a gradient. It's moving things against its will. Okay, this was best illustrated in our most recent slides by the proton pumps in the electron transport chain. They were moving hydrogen ions, just like in this diagram, across the membrane into a space of higher concentration. Instead of using ATP, they were using high energy electrons as their energy source, but still active transport. They're moving something against their gradient. Secondary active transport then uses the gradient of one molecule to drive the movement of another. So if you build up this molecule of, in this case, hydrogen ions or protons, those protons want to return to equilibrium. So they're going to be looking for a back door to get to the other side of the membrane. And when they do that, they can use a protein like this, which binds the protons and brings them across, and also binds in the same movement a sucrose molecule. So the sucrose molecule is really piggybacking on the movement of hydrogen. Hydrogen is coming down its gradient. Sucrose is along for the ride and is being transported against its gradient. So it's using the power created by the hydrogen gradient that was originally created by the primary active transport. Okay, let me go back to this. We had, I think, a little quiz over this, I believe. The different types of ways molecules can be transported. All of them include the important concept of the conformational change, right? Which is that a protein can be in one shape or another, and when it changes shapes, it can do some type of work. So the work that this protein does when it changes its conformation is it moves things from the inside, or the outside in this case, to the inside, right? These are molecules that can't cross the membrane. And so when they bind this hydrophilic pocket, that's their little safe place. And when the hydrophilic pocket then faces the inside, they can move into the water, which makes up the cytosol. Okay? This can happen in a number of ways. We can move one thing in one direction, like the former singing group. In this case, it's a uniporter. we can move two things in the same direction, which is a symporter, and we can move two things in opposite directions, which is an antiporter. Okay, these are just terms you'll want to commit to memory. How are we doing? Still on board, okay. I tell you about the semester when One Direction broke up. It really hit some of our students hard. And I tried not to say the words One Direction, which is hard to do in a science class. There's so many things where you say, this is happening only in one direction. DNA synthesis occurs only in one direction. Symporters are moving molecules only in one direction. But I had to come up with synonymous ways to say that because I had one student in particular who was really in a bad place. And every time I said, like uniporter, emotions flared. This is a true story. Okay, where are we at? I just needed a moment. Diffusion concepts, cells, shrinking membrane proteins, taking up solids or particles in bulk amounts. This is pretty straightforward. It was at the end of this slide set. And when we take up solids in bulk amounts, we don't think of moving them across a little transport protein. Instead, we grab them up in a vesicle, which is a circular structure made of phospholipids, and that vesicle can move things throughout the cell. Okay, it's kind of like a little safe space in which we put cargo. And so in this case, the cargo is moving out of the cell, right? It's coming from the Golgi. We think of the Golgi as the shipping center of the cell. So once something gets to the Golgi processes it, packages it, and then takes it somewhere. It doesn't always take it out of the cell. It does in this example, but it doesn't always take it out of the cell. Sometimes it moves it from one place to another within the cell. And so using those vesicles, exocytosis means bulk transport out of the cell. Okay, so the vesicle gathers around the cargo, it gets to the membrane. Because the vesicle and the membrane are made of the same thing, when they fuse together, the vesicle becomes part of the membrane and opens up, and out comes the cargo. Okay? Now, if we go in the other direction, you can actually grab things from the extracellular environment, either using receptors, or sometimes you just invaginate around it, which means to capture it and form a vesicle. So this is like putting the previous slide in reverse motion. You're bringing the vesicle in instead of moving the vesicle out, but the same concept. Once that comes in, we call that endocytosis. So this is bulk transport into the cell. And if we move things into the cell, once they get into the cell, lots of different things can happen. One example in this slide is that it fuses with a structure we learned about in our cell anatomy section, which is a lysosome. This is a vesicle that contains digestive enzymes, and so you break things down, okay? But it could have other destinies as well. Now, there are two subcategories of endocytosis, which are pinocytosis and phagocytosis. Pinocytosis means we are bringing bulk transport into the cell of liquids, okay? And phagocytosis, which is like the backbone of the immune system, basically, is bulk transport into the cell of solids. And solids, in this case, includes other cells. So you could phagocytose a bacterial cell, for example. That's one thing your immune system is doing. You could phagocytose a dying cell of your own. Some of your cells, when they reach the end of their life, break down into smaller pieces, and other cells gobble them up. Okay, it's a natural process. It's a good process, and it protects you from bad cells doing bad things. Okay, what type of transport is occurring with sodium potassium? What are the steps involved? How is ATP involved? All right, last topic in this chapter. Let's go back. All right, here is the sodium potassium ATPase. You spent a lot of time on this. I'll ask you a few questions about it for sure. And you will for sure get those questions right because you're going to put this kind of enthusiasm and work into it. Imagine if we were in competition with the other 171 section. We need to come out on top. These guys are chumps. I'm just kidding. They're good kids. But what if we came out on top? I could really rub that in Dr. Foltz's face, you know. Look at my scores, man. We don't do that. We're all interested in your success. Let me retract the last 90 seconds what I said. Let's just do the best we can and not worry about being better than other people. We're all in this together. High School Musical. Full circle. Okay, so what about this one? We can conquer this just like you conquered the glycolysis family tree there. There are a few major concepts here, and if it looks complicated, put that thought out of your mind. Just say, what are the main things I need to know about this, and conquer it. So the main things you need to know, this is active transport. So it uses ATP, right? That's our energy molecule. It's moving things against their gradient, okay? And it's moving more charges out than in. So forget about the sodium and potassium for a second and just look at the plus on both sides. So 3 pluses go out, 2 pluses go in. Eventually that's going to create an imbalance, right? There'll be more positives outside than in. So that's the the electrical gradient. In addition, we're moving sodiums across and potassiums across the other way. So we're also creating a chemical gradient. So this is the first example we've seen of an electrochemical gradient. A gradient of both different atoms as well as different charges. In addition, when these things move back and forth, what you see here is the amount of work done by 1 ATP. So this is predictable and calculatable. So if somebody were to say, how many ATP would it take to move 6 sodiums out of the cell? You would say...
It's not an answer to the question.
Oh, okay, sorry.
I'm sorry, I didn't catch. How is it a chemical gradient?
It's a chemical gradient because if this was a cell where the ATPase was working, we are building up more sodiums outside than in, and we're building up more potassiums inside than out. That's the chemical gradient. Does that make sense? It's a gradient of molecules. So we could think of them individually. If you just think of the sodium, that's a chemical gradient. Just think of the potassium, that's a chemical gradient. If you don't think of either of those things and you just look at the charges, every 3 charges that go out, only 2 charges come in. So eventually, you're going to start building up a big difference. So that's the electrical gradient. Gotcha? All right. So the question was, if we want to move 6 sodium out of the cell, how many ATP would we need? 2. What if we wanted to move 9 sodiums out of the cell? Right, it's just simple stuff, right? So don't overthink these things. There are numbers, and sometimes when there are numbers, people think of math, and when people think of math, their knees turn to jello. But we must embrace math. Math is the basis of physics, which is the basis of chemistry, which is the basis of biology, which is why you're alive. So we should love the math. It's taking care of us on a deep level. Okay, now let's conquer this diagram without panicking. So what type of transport? What are the steps involved? How is ATP involved? Okay, 3 questions. Let's answer them. So the type of transport is active transport, right? Because energy is directly involved. So we have those. Oops, I'm in my no C zone there. Let me move over. So the type of transport is active transport. If we see ATP, that should set off a little light bulb. It says active transport. In addition, this is using a molecule that's moving 2 different things in opposite directions. Not one direction. Don't need to get emotional. 2 things in 2 directions. So what type of transporter is our sodium potassium ATPase? It's an antiporter. Okay, so it's an antiporter doing active transport. And the ATP is involved because the ATP is going to force the conformational change. So let's say that first, and then we'll look at how it works. So adding a phosphate from ATP causes the conformational change. This is the key to how this works. Conformational change. The conformational change means that this protein is going to change shape and have a different binding affinity when the phosphate is there than it did when the phosphate was absent. So without the phosphate, it's facing in, and it has 3 binding sites. Those binding sites want to bind sodium. Okay, when an enzyme wants to bind to its substrate, it has a very specific chemical interaction based on the structure, in this case of the sodium atom and the pocket inside the sodium potassium ATPase. So it's a puzzle piece fit. It doesn't want to bind potassium, not yet. Okay, when ATP is added, a phosphate is added to this protein, and a phosphate has charges. It's a pretty large functional group of a phosphorus with 4 oxygens. That changes things. When it binds, the conformational change flips that protein to the outward facing state. In the outward facing state, what used to be a perfectly good sodium binding site changes. Now the sodium and the enzyme don't have affinity for each other, so the sodium just checks out, okay, and moves on. And instead, now we have potassium binding sites. So the potassium now are nice and snug in that enzyme, but the trick is that when potassium bind the enzyme, they cause the phosphate to pop off. And when the phosphate pops off, the enzyme says, wait a second, I'm not supposed to be facing outside, and it flips back to its original shape. Okay, it only faces outside when the phosphate is there. So when the phosphate pops off because of the potassium binding, it flips back in. And now it's reset. So it doesn't want to bind potassium anymore. Potassium leaves. It's back to its old sodium binding ways. All right, so active transport, it's an antiporter. The phosphate from the ATP, this is how energy is put into this enzyme. The phosphate changes the shape, but that change is reversible. So this ATPase is moving between these 2 states, right? Binding sodium on the inside, binding potassium on the outside. And each time it changes, it moves those molecules across the membrane. Okay, questions about that? Can you conquer this? You can. This is going to be like in Apollo 13 when the astronauts are stuck in space and NASA says this is going to be our worst moment. And Ed Harris, who's playing the NASA director, says, I disagree.